Welcome to this episode. Today we're diving into a really interesting topic: why biomarkers might be the key to bridging the gap between a biological mechanism and the evidence needed to validate it, especially in early-stage biotech research.
Yeah, it's a fascinating area. In biotech, researchers often start with a hypothesis, an idea about how a treatment might work inside the body. But having an idea isn't enough. You need to actually measure whether that idea is happening in real biological systems.
Exactly, and that's where biomarkers come in. They're like biological signposts or checkpoints that help scientists see if the biology is changing as expected. But before we get too deep, maybe we should clarify, what exactly is a biomarker?
Good point. So a biomarker is basically any measurable biological signal that tells us something about what's going on inside the body or a biological system. It could be a molecule, a protein, a receptor signal, an inflammatory marker, or even something like an imaging result or a behavioral change.
Right, think of it like the gauges on a car dashboard. They tell you if the engine is running smoothly or if something needs attention. For example, if a drug is supposed to reduce inflammation, an inflammatory marker in the blood might serve as a biomarker to show whether inflammation is actually going down.
That analogy really helps. So biomarkers are like these biological readouts that make invisible processes visible. And that's crucial because in biotech, a mechanism, which is basically a proposed explanation for how a treatment might work, is just a theory until you can measure it.
Yeah, without measurement, it's all just speculation. Researchers need to ask questions like, is the target being engaged? Is the pathway activated? Are the changes dose-dependent? Are the effects reproducible? Biomarkers help answer these by turning hypotheses into measurable data.
And that's so important because it moves research from, we think this might work, to, here's evidence that it's working biologically. But, of course, early-stage biotech is full of unknowns. You don't always know if a candidate drug is stable, safe, or even hitting the right biological target.
Exactly. Biomarkers don't eliminate risk, but they help reduce uncertainty by providing measurable signals. Like, if a biomarker shows no change in the target pathway, that might suggest the drug isn't engaging the target, which could mean it's time to pause or refine the approach.
On the flip side, if biomarker changes are reproducible and align with expectations, that can justify moving forward to the next stage of research. It's a way to use time and resources more efficiently.
That leads us to go, no-go decisions. Those formal checkpoints where researchers decide whether to advance, pause, refine, or stop a program. Biomarkers provide key data for these decisions.
For example, if receptor engagement can't be confirmed, the program might need refinement. Or if safety biomarkers raise red flags, development might pause. But if biomarker responses are reproducible and look good, the program can proceed.
It's a structured approach that helps avoid costly mistakes and focuses efforts on promising candidates. Now, BII works on several research stage platforms, each with unique biomarker needs. Maybe we can talk through some examples?
Sure. First up is Neuroforol. It focuses on neuroinflammation and receptor-selective small molecules. Biomarker questions here include, does the drug engage the intended receptor? Are inflammatory markers affected? Is the response reproducible? And importantly, does it avoid unwanted activity, like engaging the CB1 receptor?
Then there's mycophorol, which is inspired by fungal biology and targets neurotrophic pathways, those that support nerve growth and resilience. Biomarkers might include levels of BDNF or NGF, which are proteins that promote nerve growth, receptor signaling, dose dependence, and safety markers.
That's a good one to highlight, because neurotropic claims can be easy to overstate without solid biomarker evidence, so careful measurement is key.
Absolutely. Then there's neuroreset, which is earlier stage, and focused on recovery biology after dependency, neuroplasticity, and brain recalibration. Biomarker planning is especially critical here to define measurable signals related to stress response, neuroplasticity, reward circuits, sleep, and cognitive function.
And finally, BII's precision peptide platforms raise special questions about stability, delivery, immune response, and target engagement. Biomarkers help connect the molecular design to actual biological performance.
Now, it's really important to stress that biomarkers are not treatment claims. They show biological activity, but don't prove clinical benefit.
Right. Just because a biomarker shows a pathway is activated doesn't mean the drug actually treats a disease or is safe and effective in humans. Clinical trials are still needed to establish that.
BII is careful with language to avoid overstating what biomarker data means. It's about guiding validation, not replacing it.
That's a crucial distinction. Also, not every biomarker is useful. A good biomarker should be relevant to the mechanism, measurable in the chosen model, reproducible, and connected to the target.
Poorly chosen biomarkers can add noise and confusion. Safety biomarkers are also important early on to detect risks like toxicity, immune reactions, or off-target effects.
Catching safety signals early can save a lot of time and money by identifying problems before big investments.
Biomarker planning also helps when working with partners like contract research organizations, academic labs, investors, and strategic partners. Different partners focus on different biomarker questions.
For example, a pharmacology lab might focus on receptor binding, while a safety partner looks at toxicity markers. Having a clear biomarker plan makes a platform easier to evaluate and more attractive for collaboration.
And biomarkers fit into a bigger picture that includes target selection, mechanism, biomarker plan, safety screening, delivery strategy, and validation milestones.
This structured approach helps research stage platforms develop in a clear, logical way.
So, to wrap up, biomarkers are like biological signposts that help researchers see if their ideas about how a drug works are on the right track.
They don't replace clinical trials or prove treatments work, but they're essential tools for moving from hypothesis to evidence.
For BII, biomarkers are a bridge, connecting scientific hypotheses to measurable biology, guiding decisions, and helping partners understand the potential of new platforms.
And that approach makes early-stage biotech research more transparent, efficient, and grounded in measurable science.
Thanks for joining us today. We hope this gave you a clearer picture of why biomarkers matter so much in biotech research.
Yeah, thanks for listening. Take care.